Hi, everyone. Thanks for joining the showcase session. In this session, the goal is to give you guys an opportunity to meet the experts that are using Nanopore technology for carrier screening in some of the genome's most challenging genes. I'm going to introduce each speaker, and they'll talk a few minutes about the work that they're doing, and then we'll have a chance for Q&A and a general discussion. One housekeeping note is that this is an on-site only event, so we won't be taking questions from the app. If you have questions, please raise your hand and we have a microphone that they can bring to you. Our first speaker is Dr. Brad Hall from Asuragen. Brad received his PhD in molecular biology from the University of Texas. During this time, he developed the Aptamer stream within the Freshman Research Initiative, the largest undergraduate research program in the United States. He then joined Altermune, founded by Nobel laureate Kary Mullis, where he focused on immune redirecting therapies. Since 2016, Brad's been with Asuragen, where he's now the Director of R&D. Asuragen has a long history of developing tools for some of the genome's most challenging genes, and Brad is leading the transition of these assays over to the Oxford Nanopore technology. Brad?
Thank you. Thank you very much. Hi, everyone. Thank you for coming. Thank you for listening. Getting a little feedback. Okay, cool. As Alex mentioned, I'm helping to develop the newest assay that Asuragen is going to release. And it's an 11-gene panel towards carrier screening, with an indication towards carrier screening. The 11 genes are traditionally very difficult to detect variants in carrier screening, by short read sequencing. And so we've utilized our AmplideX PCR amplification technology and also long read sequencing with Oxford Nanopore to come up with a kit that will hopefully address some of these genes like FMR1, SMN, SMA for SMA, CYP21A2, GBA, F8 intron inversions, the thalassemias that have HbA1 and 2 HBB, so that is what I'm involved in for the last 2 years, and we've been working with colleagues recently within the past 6 months to test out this assay on clinical samples.
I'm going to ask you a lot of questions about why they're so challenged.
That's fun.
Thanks, Brad. Next, I'd like to introduce Dr. Anne-Sophie Lebre.
Yeah.
I did that terrible one, sorry. Anne-Sophie is a full professor in human genetics at Reims Medical School. I would have pronounced that Reims. Reims. I'm getting better. And is a clinical molecular geneticist at Reims University Hospital. Renowned for innovative contributions to DNA sequencing and epigenetic studies, she drives forward both scientific knowledge and practical applications. Professor Lebre's work enhances the precision of genetic analysis, translating discoveries from bench to bedside. She's particularly interested in genomics and epigenetics of rare diseases and CNS tumors. In addition to her expertise in genetics, Anne-Sophie is an expert in the clinical implementation of genomic technologies. We are honored that she is applying Oxford Nanopore technology to her work.
Thanks for the invitation. So, I'm really into point of care and fast sequencing, and so that's why I'm really interested in the Asuragen kit. We met at the London calling 2 years ago, and I came to see Brad. I want to test your kit, and then I became the beta test site for France and then for Europe, right? And so now we are up to 250 patients, and we tested all the mixes, and we collected patients from all around the country, and we got very challenging diagnostic, and it seems to be really, really nice. And now we are going to, we were in Minayon, and we are jumping to the Flongle because we want to have some fast track analysis. And we got SMA diagnosis 2 days ago, and the bioinformatics took us 6 minutes for 2 patients, so it's really fast. So I'm quite happy with this test, and we can discuss about it a bit more.
Wonderful. Thank you. Let's take a seat. I have a couple questions for you, and then we can take questions from the audience.
Yeah.
Brad, maybe first you can talk about what do people do today to get to these particularly challenging genes, and how is the AmplideX Nanopore kit that you've developed going to change that dynamic?
Yeah, thanks. For carrier screening, there are a number of genes that are traditionally easy to hit by short-read sequencing. And then there are a handful of genes that are very difficult. And these usually have large repeats that short-read sequencing just can't address effectively without extremely complicated methods. Structural variants that are much larger than a 250 base pair read, or they have copy number variation with pseudogenes or additional genes that are nearly identical. And so you got to look at that with SMN1 too. So there's lots of different genomic variation that are complicated to address by short-read sequencing that long-read sequencing addresses just fine. And we have combined some of the highest proportion of carrier screening genes along with some of the most complicated ones in that 11 gene panel that I described, to kind of give labs the opportunity to have a one-stop workflow for the most needed variants.
Thanks, Brad. Anne-Sophie, today, do you just not sequence these really hard genes, or do you use different methods, and how are you hoping that the Nanopore-based method is going to change what you're doing in the clinic?
So for X-Fragile, we were using the regular Asuragen kit with PCR-CE, and it would be nice to go to Nanopore sequencing. For the other gene, we were sending them around in the country because we are very specialized in France. Everybody is not doing everything, so we have network to send the patient. So I think it's very complementary with the other approach. We are using a lot of exome analysis, but sometimes we need some very specialized analysis, and that's why we are really interested in these 4 mixes. And we will use, according to the patient, we will play with the different mixes. As you know, maybe in France we are not using a carrier screening approach. We don't have the right to do that, but we will use different mixes according to the patient and the clinical data.
Yeah.
And I would say that most of these genes, we've needed to use multiple orthogonal methods, whether it's PCR-CE or MLPA or Sanger sequencing or something else, multiple orthogonal methods to get at the same data set the same data that we can get in this one workflow.
For each gene?
For most of the genes, yeah. For at least 2 orthogonal methods for every gene we have.
And now you're taking all those genes and combining them on one single panel.
And one workflow.
You mentioned turnaround time, and that you were able to get the bioinformatics done on these samples in just a few minutes. What was it taking you before?
I have no idea, because for SMA I was not doing the test. So the idea is to be able to take care of all patients in all hospital. Newborn screening should find some new SMA patient, but we will have to confirm them, and it can be really fast, really fast.
Yeah.
Yeah, I can tell you for SMA, copy number gene, the bioinformatics for PCR-CE assay, which is typical, or maybe a droplet digital assay. The bioinformatics are not that hard when you're just looking at copy number, but we can incorporate SNVs into that, and that would take much longer, and you do both of those independently, and it could take, multiple weeks to get all that data back and assembled into one file.
Got it. Can you each talk about how many samples have you run, and what's the performance of the of the assay and the work that you've done so far?
I'll let you start.
Okay, so we collected 250 samples from for each mixes. We have 4 mixes. One mix A for SMA and CFTR, one mix B for Asuragen, mix C for hemoglobinopathy, and mix D, it's CYP21A2, tenascin, challenge genes, and GBA. And we collected samples for all this disease, and it was very powerful. What do you want to know?
And you ran, so you've run 250 samples in total.
Yeah.
And how does it compare to the conventional methods that?
Oh, we didn't do it before because we didn't have the panel for that. We didn't order the panel. We are just performing exome, and it's very cheaper than exome analysis. It's faster, and it's very good accuracy. So you can answer very fast and with an accurate diagnosis. Okay, I know. Nanopore is not for diagnostic, I know. But it's a good way to translate it to the patients, to the clinics, and to the community, because the idea is to share that with a French colleague to help them to implement new techniques in their lab.
Yeah, so, carrying on what Anne-Sophie said, the partnership that we put together at London Calling like last year, basically she was pulling in samples from all over France across the 11 genes that we had on the panel. It was a wealth of great data for us to be able to optimize our algorithms, to be able to make these calls with the raw sequencing data that we're getting out of it. Tested 250 samples. We have other partnerships with other labs. And I think on clinical samples, when I last checked, we were over 500 clinical samples. And I think 350 of those unique. And that's clinical samples alone. We're well over 1,000 samples with cell lines and other presumed normal cohorts. So again, with carrier screening, you want to make sure that you can identify and differentiate wild type samples from samples that are either have a single variant and they're carriers, or affected have 2 variants at the same gene. And so we needed to know that we could differentiate each of those. I think we're well over 1,000 samples right now tested before going to market, and it's an RUO assay. Most labs don't develop RUOs and test them on that many samples before going to market. So we have a lot of sample sets that we've tested.
Thank you. Let me stop hogging all the questions and see if there's any questions from the audience. I'd have many more questions. Uh-oh.
I have a question for the link variants for SMA. Are you testing for the link variants?
The what variants?
The SMA 1 and 2, they have a link variants for silent carrier.
Yes. So there are certain SNVs that suggest silent carrier, so 2 copies of SMN1 on the same allele. And within our amplicon, this is a targeted panel, within our amplicon we can identify both of those on the same read that we're getting. The C or T variant denotes typically SMN1 or SMN2. And we can also detect there's a disease modifier in SMN2 that will effectively turn SMN2, which is not very functional in making proteins, into a more functional protein, and it will recover if you have a low copy count of SMN1. And so we can detect that also on the SMN2 allele. For SMN, the kit supports full exon coverage, and then we can phase variants using 2 of the mixes because we have SMN in 2 different mixes. So we can phase variants from exon 3 to exon 8 in the second mix once we get copy number in the first mix. And 95% of the variants in SMN are copy number related, but the other 5% are SNVs. And of that 5, most of them are within exon 3 and 8.
Thanks, Grant.
Mm-hmm.
I'll continue with my questions then. You've spent years working on really challenging genes. What's the gene that gives you the most heartache, that keeps you up at night? And why?
I'm going to answer that in 2 different ways. We got a sample from Anne-Sophie's collection for GBA, and it had a structural variant that we hadn't seen before. And with GBA, most of the disease is associated with SNVs, but there are structural variants because it's really close in space to that's pseudogene, about 5 kb away. And our algorithm just didn't know how to detect it, but we could see it in the raw sequence data. And so when I say twofold, really it's getting enough clinical samples to develop the best algorithms we can to translate that raw data, that signal coming out, into a variant call. And so that's what keeps me up at night most of the time. Our bioinformaticians and our software group are working day and night to get that better and better and better, but we are limited by the samples that we can test and the variants that we can see. So that is what keeps me up at night.
And Sophie, do you have a particular gene that
I'm thinking about
that is your most challenging to deal with?
I'm thinking about the next kit you should develop. Because we should think about hypomobility, neonatal hypomobility with Steiner, Prader-Willi, and SMA, and we are very close to be able to do it. So that's my suggestion now, to go further and have a 3 tests in the same tube, and to be able to answer to the maternity in a few hours should be really, really good.
Yeah, and I will say, now that we've broken into the world of long-range sequencing and we're coming out with our first kit, our hardest job is limiting ourselves to what's next, because we want to work on everything, because we're nerdy scientists. We want to work on it all. And so it's identifying the next assay that we want to come out with, the next kit that we want to come out with, the one after that, the one after that, the one after that. And so talking with people like Anne-Sophie and other customers, we get an idea of what they're interested in, what's really complicated for them, and what they want to work on.
Great. Thank you. With just a couple minutes left, I have one question, unless there's more from the audience. Okay, I will.
One quick question. Do you have the F8 inversion 22 in your 11 gene panel?
Yeah, the panels, both intron 1 and intron 22 inversions. Yep. And we detect both wild type and the inversion, so we can get zygosity.
Hemophilia, F8 inversion. Maybe you can just describe to the rest of the audience, just how hard that one is.
Well, we don't have a very good orthogonal method for it. There is a gap PCR method that's in the literature, but it's difficult because the inversion is large. And the size of fragment that we need to detect it is large. So we need at least like an 8 kb fragment to be able to detect that inversion effectively.
And that's what you do.
Yes. Yeah.
Wonderful. Any last things you want to say?
I don't know. No.
We really appreciate you guys taking the time to come here, taking the time to do the work that you're doing on our platform, and sharing that with us. Thank you very much.
And thank you.
Thank you.